Hello everyone, welcome to today's session. Today we are going to see class 12 biology chapter 4 principles of inheritance and variation. In that we are going to see mutation. So let's start with the topic. So what is mutation? Just imagine you are writing an important sentence in a notebook. If you accidentally erase a word, add an extra word or change one letter, the meaning of the sentence changes, right? So in the same way, if the DNA sequence changes, we call that as the mutation. So mutation is the permanent alteration in the DNA sequence. So since this DNA carries all the genetic information of an organism, even a small change can affect how the organism looks or functions. So a mutation can produce changes in its genotype as well as phenotype. So genotype, what happens? The genetic makeup of an organism gets changed. And also the phenotype, the observable characters like appearance or functioning, yeah, it changes. So mutation is one of the major source of genetic variation. So along with the recombination, what is recombination? We have already seen in our previous videos, right? So recombination is mixing of genes during reproduction, and the mutation, it introduces new variations into a population. So these variations are important because they provide the raw materials for evolution. How this mutation occur? So each chromosome contains one long continuous DNA molecule, right? Yes, DNA molecule, and that is tightly coiled inside the cell. Since these genes are located on the chromosomes, any change in the DNA can also affect the genes present on the chromosomes. You should remember the recombination is like a reshuffles existing genes, whereas the mutation actually changes the DNA sequence. So what, for the exam point of view, you should remember mutation changes DNA, that is the genotype also changes and phenotype may also change. So next we are going to see changes at the chromosomal level. So chromosomal mutation. See, sometimes mutations affects not only just one DNA base but a large segment of a chromosome. There are two main possibilities. So first, let's see the first one is deletion. So what happens in deletion? See, deletion means loss of a segment of DNA. Deletion means loss of a segment of DNA. Just to think of a book with one entire page is missing. Yes, so some important information disappears. So similarly, when a part of a DNA is lost, the genes present in that region are also lost. For example, let us see this one. Yeah, segment loss with gain. So the normal. So the normal one, it contains 1, 2, 3, 4. So after deletion, what happens? Yes, the second segment is lost. And next we are going to see insertion. Insertion or duplication, right? So insertion or duplication means an extra DNA segment is added. An extra DNA segment is added. So just imagine copying one paragraph twice in a book. So what happens? The information becomes repeated, right? So same thing happens here also. Let's take this normal one. So when you say here the normal one is 1, 2, 3, 4, right? When it is duplicated, it becomes like 1, 2, 2, 3, 4. So here again, the segment 2 appears twice. So, but what happens because of these changes? See, genes are located on chromosomes. So, deleting or adding chromosome segments changes the normal arrangement of genes. So, these structural changes are called as chromosomal aberrations. They are called as chromosomal aberrations or abnormalities. So, this chromosomal aberration, they often disturb normal cell functions, and they are commonly seen in cancer cells where chromosomes become abnormal. So, how to remember, like a easy analogy I'm telling you, just to think chromosome as strain and gene as compartments, right? And the deletion is one compartment is removed and duplication is one compartment added twice. So the train still moves, but it does not function normally. So exam point of view, what you should remember, chromosomal aberrations are commonly observed in cancer cells. Yeah, very important. Always remember this. So next we are going to see point mutation and a frame shift mutation. Not every mutation changes a large chromosome segment. Sometimes only one DNA base changes, right? So this we called as the point mutation. Yeah, that we called as the point mutation. So a point mutation involves the change of a single base pair in DNA. So even though only one base changes, it can completely alter the protein produced. Let us take sickle cell anemia. Yeah. So here it is, children. Sickle cell, a classical example. Yeah. Sickle cell anemia. So normally what happens here, the beta globulin gene, it contains the codon. Here you can see the codon GAG. So this codes for the amino acid glutamic acid. So this codes for the amino acid glutamic acid. So due to the mutation, what happens? One base changes. That is yeah, you can able to see here A. Yeah, it is yes, it is turned into, is changed into T. So due to mutation, one base changes. That is you can see here GTG, right? So now the amino acid becomes valine. First it was glutamic acid. Now it becomes as valine. So just this single change, it causes the hemoglobin molecules to stick together. So making the RBC becomes a sickle shaped instead of round. So these sickle-shaped RBC, they carry oxygen less efficiently, and what they will do, they just go and block the blood vessels, and it causes pain and anemia. So this shows how one tiny DNA change can produce a serious genetic disorder. So next we are going to see frame shifter mutation. Sometimes DNA gain or loses one or more bases. So this is called as the insertion or deletion at the base level. So since these proteins read in groups of 3 bases, adding or removing one base, it shifts the entire reading frame. So this is called as the frame shifter mutation. You can able to see here. So ATG, we used to read like 3, right? 333. You can see here, yeah. So what happened? One X is inserted over here. So 3 3 if you read means, yeah. I'll explain you with a simple example. Just take this one. Yeah, take this sentence. The cat ate the rat. Just remove this T. We are going to delete the T. Next, what happens? Automatically, yeah, this the 3, this 3. You can able to see. Can you can you read this out? Is it looking like a sentence? No, no. Heka, ata, teta, har, at. No, right? So now the whole sentence becomes meaningless. So similarly, the entire protein sequence changes after the mutation. And what's the difference between this point mutation and frame shift mutation? See, in point mutation, one base changes, and also the reading frame, it remains the same, right? But in frame shift mutation, what happens? Base is inserted or deleted, and the entire reading frame is changed. So exam point of view, you should remember point mutation is one base change and frame shift mutation is whole reading frame shifts. So next we are going to see mutagens. So what are these mutagens? See, mutations sometimes occur naturally. Yes. And however, many mutations are caused by external agents. Yeah. They are called as mutagens. So mutagens are physical or chemical factors that increase the rate of mutation. So they damage the DNA and leading to changes in the genetic material. So types of mutagens. So physical mutagens. So these include the ultraviolet. Yes, ultraviolet UV radiation, ultraviolet radiation, and also X-rays and also gamma rays. So among these, UV radiation is the most commonly that is mentioned in your NCERT book. So excessive exposure to sunlight, it allows the UV rays to damage the DNA. What it does? It damages the DNA and also increases the chance of mutation. Next is the chemical mutagens. So certain chemicals can also damage the DNA. What they do, they just alter the DNA bases or interfere with the DNA replication and also producing the mutations. So the detailed mechanism is beyond the class 12 syllabus. So, but remember that the chemical mutagens also induce mutations. Yeah, what is the importance of this mutagens? Mutagens increase the genetic variations. Mutagens, what it does, mutagens increase the genetic variation. And also, while some mutations are harmful and cause diseases, other may be beneficial and contribute to evolution over long periods. So, exam point of view, remember UV radiation is a physical mutagen. Yes. So, let's have a quick revision. So, mutation. So mutation is a permanent change in the DNA sequence that alters the genotype and may also change the phenotype. So along with recombination, mutation is a major source of genetic variation. So when large DNA segments are lost or added, yes, or added, that is insertion or duplication. So chromosomes become structurally abnormal. So these are called as chromosomal aberrations. So which are commonly observed in the cancer cells. And then a point mutation, it changes only one DNA base pair. Okay. Then sickle cell anemia is a classic example where a single base change, that is GAG, yeah, it turns into GTG and changes the glutamic acid to valine. And a frame shift mutation occurs when bases are inserted or deleted, shifting the reading frame and altering all downstream codons, right? And then mutations can be induced by mutagens, so which are physical or chemical agents. And UV radiation is the important physical mutagen mentioned in your NCERT book. Yeah, watch the full video, children. Don't skip the video. Yes, we'll see you in the next video. Thank you.